today we are going to talk about neuromuscular junction. What happens after the depolarization of muscle cell, right? Quickly, we will take only one vesicle from neuron terminal, and we'll see that what happens after depolarization of that muscle cell, right? So we will take one muscle here. This is one muscle fiber. We have a lot of wasting fibers, muscle fibers. This is one sarcolemma or sarcomere, right? Here we have discussed that sodium well goes inside, and after depolarization what happens here we will discuss now today, right? So we will take one neuron here from the ventral horn of spinal cord. For example, this is spinal cord, and this is the ventral horn. From ventral horn, the motor neuron, which is also known as alpha motor neuron, it arises and make synapse with this muscle cell, right? We have discussed that by depolarization the impulses are transmitted to the axon terminal, neuron terminal, and here we have a vesicle having a lot of important proteins like acetylcholine receptors or channels, and also have proton pumps, right? From GIT we are taking choline. Choline enters into neuron by choline transporters. I am mentioning these choline transporter because some of the drugs we are using in daily life can block these channels and affect muscles, right? Also bacterial toxins, viral infections, the DNA mutations can mutate this channel and it will affect the muscles, right? The next thing is mitochondria, which is present here, coming from cell body, right? And it giving acetate to the axon terminal. Mitochondria we are taking acetate, and here one enzyme is very important, is working, which is called acetylcholine transferases. Convert this acetyl and choline to acetylcholine. For example, these both combines and forms acetylcholine, and the enzyme is working here is called choline acetyl transferase, right? So for example, these are that acetylcholines forming from acetate and choline by this channel. This is acetylcholine transporter, this is choline transporter, only choline transporter, and this is acetylcholine transporter, and this acetylcholine transporter can be blocked by some medicines or some drugs and some toxins, and can affect this muscle and can paralyze the muscle, right? So for example, these acetylcholine goes inside these vesicle. From where these vesicles came? These vesicles are coming from cell body, and acetylcholine is forming here. This is very important vesicle, very important, and having important protein. So it is not something to waste here, right? It will be restored by calithrin proteins, right? So these green are now, for example, these are acetylcholine, right? And this channel, this is used by for hydrogen ions, and this hydrogen ion here only regulates the entry of acetylcholine, because acetylcholine will be denatured here after used. After use, there is one enzyme working which will denature or will catabolize this acetylcholine, right? So this hydrogen is here for regulation of entry of acetylcholine to vesicle, right? Now we have an important calcium channels working here. These are, this is just the recap. I have discussed these things, right? By this depolarization, these channels become active, because these channels are voltage sensitive, so calcium channels, right? These are voltage sensitive calcium channel. So by entry of these sodiums into this exon terminal, these channels will be activated, and it will take calcium from extracellular spaces to exon terminal. Why these calciums are important here? Because the proteins which are attached to this vesicle and this membrane are calcium sensitive. Calcium is needed for activation of that proteins, and that proteins are called V-Snares protein and T-Snares protein, right? For example, we have here, I have one this protein and one is this one, right? These are called V-Snares protein, vesicular snares, right? This protein is called synaptobravin, and this is synaptotagmin. This protein is SNAP25, and this is syntaxin or synaptotaxins, right? Why these proteins are important? Because there is one anaerobic bacteria present in environment and caused muscle paralysis. When that bacteria cause infection in our body, it will affect the muscles and will paralyze the muscle, right? What is the function of that bacteria? It will cut these proteins, right? We will discuss in diseases, right? So when calcium goes inside, it came and binds here, right? And when calcium is binding here, because these proteins are calcium sensitive, so it start contraction, right? And when it contracts, this vesicle, for example, this vesicle by exocytosis release this acetylcholine into this synaptic cleft, right? If someone asks about the components of neuromuscular junction, we'd have one is this alpha motor neuron, right? The second one is synaptic cleft. The third one is this motor end plate, right? This alpha motor neuron is also known as presynaptic neuron. You can say that this is presynaptic neuron and this is postsynaptic muscle. You can also this is presynaptic component of neuromuscular junction, and this is the postsynaptic component of neuromuscular junction, and this is synaptic cleft. And synaptic cleft, these acetylcholines are released, right? Here on this membrane, for example, this is the end plate of muscle. Which receptor is present here? Cholinergic receptor called nicotinic receptors. Acetylcholine receptors, it is also known as alpha, beta, delta and gamma receptors, and it is also known as ligand binded ions channels, right? So for example, these are the nicotinic cholinergic receptors, right? And these are ligand binded, mean that it have an active site, and when some ligand, some hormones, neurotransmitters or drugs will attach to these receptor and it will become active, right? So here the activation ligand is which thing? Acetylcholine. These are acetylcholines. For example, these are acetylcholine. It came and binds here, right? The components of these channels we already have discussed in previous lecture, right? The alpha 1, the alpha 2, the beta, the gamma and delta, right? Now when these acetylcholine binds to these receptors, it become opens, and as we know that the sodium is in concentration outside the cell, right? So the sodium goes inside to muscle, right? Now you will tell me what is the resting membrane potential of muscle? Resting membrane potential. When this muscle is in resting position, the voltage here is minus 90 millivolt. By activation and summation of all these depolarization potentials, it will become minus 55 millivolt, and this potential is called threshold potential. Threshold potential. And we have to contract all the layers of muscles, endomysium, the perimysium and epimysium, so it will require more charges. These charges are not enough for contraction of muscle. So when it become, for example, this become threshold potential, right? Here we have voltage gated or voltage sensitive sodium channels on each side of this neuromuscular junction. This is the neuromuscular junction, and this is the other components of muscle, right? So these threshold potential will activate the sodium channels, right? These are the sodium channels, but this sodium channel is ligand binded, ligand sensitive, and this is voltage sensitive, and this voltage is provided by these channels, right? For example, it is in resting, for example, it is minus 90 millivolt, and when these channels activated, the voltage become minus 55 millivolts, and this is now the threshold, and by this potential these gates will open, right? So sodium will rush into muscles, right? And it start depolarization. Now for example, here this potential is depolarization potential, right? And this is now, for example, this is 30 millivolt, plus 10 to plus 30 millivolts this depolarization potential will reach, right? So now we will discuss the structure of this thing. We have discussed already the structure of this, that it has 5 units further: the alpha 1, alpha 2, beta, delta and gamma. What is the structure of this channel, it is just like, for example, this is the voltage gated sodium channel, right? It have two components here. One is called the activation gate and the second one is called inactivation gate. As we have discussed that it have inhibin protein like this, inhibin protein, and it blocks this channel. When ligands came and binds here, these inhibin protein become like this and this channel become open. Here it have one activation gate. This is, for example, activation gate, and this one is inactivation gate, right? So when this threshold potential reach here, this activation gate become open abruptly, quickly it opens, right? This is the activation gate. Activation gate, and this thing is an activation gate. Because why these both gates are important, are required here. If this threshold potential came here and activate this channel, and it activates for a long time, by long time this activation will block this channel and no more sodium will able to come inside to muscle cell, right? So for this, activation gate opens abruptly. For example, this is activation gate here, and when threshold potential reach, it become open, and when it open, the sodium rushed into the cell. This inactivation gate closed very slowly, right? For example, this is the inactivation gate and this is the activation gate. When this open, it will move very slowly, right, and will block this channel, right? So for example, it become open and the sodium is coming, and this channel, this gate is moving in this direction, and last for example, this is that activation and this is inactivation. This inactivation gate came and block this area, right? Gorai, the activation gate is the zirculavi. When this chain is activated, it become depolarized, right? What is this component is called? This is called triad. This is called triad, right? For example, we have this is, for example, endoplasmic reticulum. Here it is called sarcoplasmic reticulum, right? And here is also one endoplasmic reticulum component is present, right? Here also these are right, and the function of sarcoplasmic or endoplasmic reticulum is the storage of calcium. So a lot of calcium is stored in this sarcoplasmic reticulum, and our target is to release these calcium for muscles contraction. So this depolarization is required for activation of this. Why? Because there are some important proteins. For example, one protein is standing here and it holding in its hands another protein. This is, for example, the proteins he is holding in hands, right? What is the name of this protein and what is the name of this protein, right? This green protein is called dihydropyridine protein, right? This protein is called, this is called hydropyridine protein, right? And what is the name of this? This is called ryanodine proteins. Ryanodine proteins, and these proteins are also depolarization or voltage sensitive proteins, right? For example, he is holding this protein in hands, and when depolarization came here, it will contract this protein and will make a hole in this endoplasmic reticulum, right? For example, I am taking this protein here. For example, he is standing here, and this is the hands of this protein, right? And he is holding the ryanodine protein and right, this is ryanodine protein and this is dihydropyridine proteins, right? When this membrane gets depolarized, it will shock this protein, right? And he will contract this protein with its hands, right? And now when he contract this protein, a hole makes here like this. For example, he contract this protein, and this hole is responsible for releasing of this calcium from endoplasmic reticulum. Now calcium is coming to the muscle cells from endoplasmic reticulum. For example, this is the sarcoplasm, or this is called the cytoplasm, and this is the sarcoplasmic reticulum. And by making these holes, this calcium release into the sarcoplasm or cytoplasm, and now this calcium is freely floating in sarcoplasm, right? Why this calcium is so concentrated in endoplasmic reticulum, not in cytoplasm? Anyone? Why is cytoplasm केवल इन स्टोर की? Mean there is something which is responsible for this storage, right? And a lot of proteins present inside here. For example, these are the proteins present inside. Do you remember the calmodulins? What is the function of calmodulins? The release of calcium. And what is the function of this protein? The holding of calcium with itself, right? And these proteins are called calsequesterine proteins. These are called calsequesterines, and they are called calmodulins. Calmodulins are responsible for releasing of calcium from this sarcoplasmic reticulum and endoplasmic reticulum, and calsequesterine is responsible for holding this calcium inside, right? So when depolarization came here, this calcium comes out to cytoplasm. What he is doing in cytoplasm? Just remember, this is the myofibrils are present in this cell, right? For example, this is the Z line and this is the Z line, right? And this is actin proteins. These are the actin proteins, right? And actin proteins having the troponin. Troponin have three sites: a troponin I, troponin C, and troponin T, right? One is holding the tropomyosin, the second is actin, and the third one is calcium. So for example, these are the troponin. This is calcium site, this is actin site, and this is the tropomyosin site, right? And these are the heads to which, these are the sites to which the heads of the myosin will attached. And I have told you that these sites are blocked by tropomyosin, right? So for example, this is the troponin, and troponin holding tropomyosin, and tropomyosin blocks all these sites. And now the myosin heads, for example, these are the, this is the myosin, and this is the myosin, and these are the myosin heads. Right, these are the myosin head, and these are not able to binds to actin, because tropomyosin blocks the sites for the heads of myosin. These are tropomyosin. This red protein is troponin, right? Troponin have three sides. This is the troponin C, this is troponin I, and this is troponin T, right? Now this calcium, when came out from sarcoplasmic reticulum, it will goes and will binds to this site. For example, here is on each we have troponin C, this is the troponin C, and calcium binds to all these troponin C. What is happening when calcium binding to this troponin C? It will activate this domain like receptors. When calcium bind to its site, it will activate this site, and when it activates, this will bring the conformational changes in this tropomyosin, and now these sites are available for attaching of myosin head, and myosin head will goes and will bind to these sites, right? There is one cycle or mechanism behind this contraction we will discuss now in just a few seconds, but this calcium is responsible for attaching these myosin head to actin proteins, right? Now it have some functions of ATP here. When ATP binds to this head, it will inactivated, and when hydrolysis take place, the ATP will break down into ADP and phosphate, and it will bring some conformational changes in the head of this myosin. And after the detaching of phosphate head, a phosphate group from myosin, it will attach to the active site and will contract, right? We will discuss that cycle, but here one another example. For example, this is the myosin, right? And this is the myosin head, right? And this blue protein is actin, and we have these are the sites for binding of myosin heads, and this green protein is tropomyosin, which blocking these sites. and this green protein, which is called tropomyosin, is holding by troponin specific site, right? This protein is troponin. When this calcium came here, it will bind here and will bring some conformational changes in this tropomyosin, right? And this head is now able to bind to these sites. This is the story for this calcium. Now, when this happens and the muscles contract, where this calcium will go? They are not wasting, right? Because this endoplasmic reticulum have reuptake channels, having reuptake channels. This is called the calcium channels, calcium reuptake channels. And when these calcium detach again from these sites, troponin C, it will goes again to endoplasmic reticulum. Some of the channels are present here. These are also calcium channels, and some of the calcium will goes outside, right? Here, now the target is to bring these muscles again into resting membrane potential. Why? Because if this depolarization potential is remaining here, the muscles will be in contracted position. It cannot now able to relax, right? After repolarization, it will relax, right? So for that, we have channels here called the potassium channels. Potassium channels. These are, for example, potassium channels, or these are the potassium channels. And potassium is in concentration inside or outside? Inside. So in competition of these sodium channels, the potassium channel well opens, right, after depolarization, and now the potassium is leaving the cell and it brings the negativity inside the cell. And this graph will, for example, this is the depolarization peak potential. It is also known end plate peak potential. This is called end plate peak potential. When this muscle is fully excited, right? Now when these potassium channel opens and also this calcium is leaving the cell, right? Also this is when the calcium coming again into this sarcoplasmic reticulum, it brings negativity outside, it brings negativity outside. So this graph will drops to threshold and will to resting membrane potential, and now in this the muscles again relax and is ready for another cycle, right? Now after this we will discuss the contraction mechanism in these muscles, right? Because we have some bands here. This band is called from here up to here. This is called A band. This is called A band, and this is called I band. I band, and the band from here up to here, this is called H zone. H zone, and we have some proteins here, the myomycin proteins, which are making this M line. So in contraction of muscles, we will discuss that what are the changes occurs in these zones, right? Which zone will disappear, which will contract, and which zone is or which band remain the same during muscles contraction, right? په دې باندې پوه شوي؟ دا خو ډېرې پکې زړې خبرې